All right, so right in here, we have our ingredients for our extraction. We have salt, we have alcohol, we have soap, dish soap, Palmolive. Not I'm promoting the brand, and we also have water. Water, the source of life. So what we're gonna do is just step by step. We're gonna see how we extract its DNA. Let me put on a pair of gloves so I don't do a mess. And again, I just want to go to that question. I forgot who asked it, but can you extract? Can you see your own DNA? Yes, you can see your own DNA. You can see a DNA as long as there's something that is alive: a bacteria, a plant, a fungi, an animal, like yourself. You can extract the DNA because they have DNA inside their cells, or the cell if you're an animal or an organism that is made of only one cell, like bacteria. All right, so here's what we're gonna do. We have our recipient, we have our container, we're gonna add some water. In the instructions that I'm gonna post, it says 90 ml, but I don't. If you kind of measure it, just keep in mind that just take as many as much as you want, but try to keep it limited because if you have a strawberry, you don't want to use a lot of water because then it's gonna dilute too much. It's gonna be too much water for just one strawberry. All right, so we're gonna use this, and then we're gonna add 1/10, so 10%. So if this is 90% of water, we're gonna use 10% of soap. So we're gonna do it in here, 10% of soap. Let me just use some more of this. Try to be try not to put too much soap because it's gonna be very bubbly, like we did in the classroom. I think in the classroom we added too much soap, and that's why we had so many bubbles happening. And last thing we need to add right now is the salt. So just add, I'd say maybe 1/4 of a tablespoon for salt. Just add it like that. I'm gonna add a little more actually. Use your judgment. All right, so that's our solution. This is the solution that is gonna be able to break into the cells of the strawberry, or if you're doing it with human DNA, human cells, this is gonna be the solution that is gonna break into the cells. The reason it's able to break into the cells is because soap breaks fats. So when you're washing the dishes, which I hope you wash the dishes, you're breaking the fat from the dishes, right? So our cells are actually surrounded by fat. That's the cell membrane that protects the cell and protects the DNA. It's made of fat. So we want the soap and the salt to actually dissolve that membrane, dissolve that nucleus, so we can get access to our DNA. All right, so we're gonna put the strawberry inside the Ziploc bag, blah, blah, blah. And we're gonna add our solution that's gonna break into a cell. So we're gonna add it, and then we're going to just mush it, right? Oh, so we're gonna mush it. Again, a lot of you did it in the classroom. Some of you maybe tried it, didn't work. Maybe you were using too much soap. I don't want to just make too much bubbles. Try to just mush it, but don't make too much bubbles, okay? So try to avoid the bubbles because the bubbles are just gonna be soap. We don't care about the bubbles. The bubbles are just soap. So as long as you have a really good solution right here of strawberry mush juice, we should be okay. So again, right here, what we have is the cells that are being broken down by the soap and the salt. The second part is that we're gonna filter this. So you can use a filter, whatever you want. You can use a coffee filter if you feel like using a coffee filter. And then we're gonna add our mush so we can filter our juice. So we don't want big pieces of strawberry because those big pieces are gonna be cells that weren't broken down. All right, so we have our solution right here. That's good enough. And then what we have here is our cells. Right now, because they were already broken down, the DNA is outside the cells, but we also want to separate the DNA from the cells. So right now in here, we actually have our DNA that is floating, but what we want to do is a method for separate it. And how we do that is adding alcohol. So I think the best example for separation is when you mix water and oil. When you mix water and oil, they separate right away. And that's something we can use in here, but instead of oil, we're gonna use alcohol. And we're gonna see how it works. So I'm gonna grab another container. I don't need that much liquid. In fact, I actually want a container that has a small opening, so I don't get that many bubbles. I'm gonna be careful when I transfer it, so I don't get the bubbles. I really want to avoid the bubbles, and I'm just gonna keep adding. I would say add a little less than it's in the container. Again, I'm gonna send the instructions, so you can use your best judgment, but just be conservative. Ooh, a little bit spoiled in here. All right. Anyway, so we have our solution right here. Our DNA is floating. So what we want to do is add alcohol. In this case, I'm using isopropyl alcohol. It's 70%. I would say don't go below 70%. Don't go 50%, or I don't think it goes below 50%. But don't use a low concentration because you want concentrated alcohol. A lot of alcohol that is really concentrated and not diluted with water or whatever else. And you're also gonna see here that I have it in a bath. This bath actually has ice. So you don't need to have ice, but make sure the alcohol is cold. If you wanna put it in the fridge just a little bit before you do it, that's also fine. And I'm gonna add just a little bit of alcohol. You wanna add enough. So one, it breaks the bubbles. And two, you wanna form that layer on top. So I'm gonna add it right here. And you're gonna see the bubbles are gonna start breaking. And you should also see that it's forming a layer on top. So maybe you cannot see it right here, but you can see actually something is floating to the surface. So the bubbles are gone, but you also see something that is flowing to the surface. So you're gonna let it rest for a while just for you to separate the DNA. In the meantime, I just wanted to tell you, DNA again is a long molecule. So it looks like this string. And if you look closely in the string, it's separated by different colors. What those colors represent is different genes. Right? So maybe one gene is for hair color, maybe one gene is for height, maybe one gene is for something else. So we have different genes in here. And that's a good representation for DNA. A lot of you did that origami DNA. That's also a good representation for DNA. The only bad thing is that this is too short. So that's what I was telling Elisa, just paste some of these together so you have a long molecule, because DNA is super, super long. It fits in the nucleus of our cell, but it's super, super long. And the reason it fits is because it's very mushed together and it makes chromosomes, right? So it's organized into little chromosomes, just like little pockets, little books of information. All right, so we're gonna check our DNA here. I actually did another one right before we joined this class because I'm nervous and you never know if something is gonna work when you do it the first time. So you wanna do it at least 2 times. And we're gonna see in here what's happening. All right, so we have in here, I'm just gonna use some tweezers. These are lab tweezers, so I used to use this in the lab. And you're gonna see in here, ooh, I get this bubbly thing here. I don't wanna spill it in my computer, but you see this mushy thing in here. It fell on my keyboard, by the way. This mushy thing is actually the DNA we just extracted. It's clear, so DNA doesn't have a color. The reason sometimes you can see it with color in some images or even some videos is because they add dye to it. Maybe not food dye, but they add some dye that you could see it in the microscope. When we were looking at cells in the microscope, we would also add that bromothymol blue or other color that we could use to see the cells, because cells are super clear. They don't really have a color. So right in here, you can see the DNA. Maybe, is it gonna focus in here? Obviously, it's a bunch of DNA together from thousands and thousands of cells. So that strawberry we have in there, that strawberry was made of millions and millions of cells. So this DNA is just the DNA of a lot of thousands or even millions of cells.